Dan Gretler. I'm a consultant for biotech companies. I'm not an employee of COA. Thank you for giving me this opportunity to talk to you about BBP 671, and thank you for the t-shirt. I was thinking of asking the organizers for an extra 30 minutes to show you the slide that the lawyers snuck into my presentation, but I decided against it. Oh, sorry, yeah. I decided against it. So I'm a physician. I consult for small companies, as I just said. I get paid for that. That's my real job. That's my daytime job. I don't see patients anymore. And the other disclosure is BBP 671 is a investigational drug. It has not been approved anywhere in the world. It has not been shown to be safe and effective for any indication, including PKAN. Of course, we're trying to change that. The agenda today, I'll talk to you a little bit about BBP 671, although Susie did a wonderful job at introducing it already. I'm going to talk to you about the first in-human study that we did, and then tell you a little bit about the plans for studying this in PKAN patients. Let's talk about COA. It's the name of the company, but more importantly, it's an important helper molecule that's in our bodies. It helps a lot of the enzymes that are present in our body. I was curious, I Googled it last night. Apparently, according to Google, we have 75,000 different enzymes in our bodies. It's got to be true. I read it on Google. COA is important for the metabolism and for helping these enzymes do their work. How is COA made? You heard from that. Oh, oops. How do I go back? Okay. So COA is made from a vitamin, B5, pentophenate, and it's made by enzymes called PANK, pentophenase kinase, as you just heard. There are ways to bypass PANK by using a later compound, as you heard from the CoA-Z compound. But I'm going to talk to you about the PANK itself. The only thing that is amiss in PKAN is that this enzyme PANK2 is not functioning properly, and so it's not producing enough CoA. The reason is, as you know, there's a genetic mutation in the recipe to make PANK2. And so the PANK2 that results from that faulty recipe is just not working well, doesn't make enough CoA. As Susie said, there are other PANK enzymes. That's the good news. There's PANK1, PANK2, PANK3, but they don't automatically compensate for the lack of PANK2 activity. Because they're not sort of linked to PANK2, there's no feedback. They don't know that PANK2 is malfunctioning and that there's not enough CoA. But this is where BBP 671 comes in. Because BBP 671 stimulates these PANK enzymes, all the PANK enzymes. And the idea is by stimulating especially PANK1 and especially PANK3, it can make up for the deficient CoA production that is due to the not working PANK2. So that's the idea. And by increasing CoA, since that's the only thing that is amiss in PKAN, this could improve the clinical picture and the manifestations of the disease, especially if we start using it early. Let me, please ignore the left side for a minute. We're going to talk about the right side, because when you have a new molecule that's been made in the lab, it doesn't occur in nature. No human body has ever seen that molecule. We don't know what's going to happen. Is the hair going to turn green? I mean, we don't know, right? So there's 5 questions we need to answer with pretty much every new drug that we develop, and this is what I do for a living. I answer these 5 questions all the time. The first question is it tolerated? If I gave you 20 milligrams of stuff and you start vomiting, or I take 30 milligrams and I start having horrible headaches, it may not be a safety issue, but it's not tolerated, and so it would be a non-starter. The second question is safety. The lawyers wanted to take me to take this out, and they changed my 5 questions to 4 questions. And I had to fight back because, well, their point is you can't do a first-in-human study and they say, aha, the drug is safe. That's not how it happens. You need a phase 3 trials, you need efficacy and safety so you can weigh the risk-benefit ratio, and that's really when you can say the drug is safe for the intended purpose. So they're right in that sense. But I'm also right because we do look at early safety signals as early as in the first-in-human study, and I'll show you an example of that. I mean, if the liver starts acting up or the kidneys start shutting down, even in the first study, well, we need to know that. The third question is pharmacokinetics. When you swallow a drug, does it go into the blood? Well, some drugs don't. That's why you have to inject them. Well, if you're going to take a drug for the rest of your life, every day, it might be nice not to inject it, right? Although diabetics do it, but still would be better to have an oral drug. So is the drug orally bioavailable? Does it get into the blood? Can you take it with food? Some drugs, if you take it with food, they don't go into the blood. And then how long does it stay in the blood? You're familiar with the term half-life, right? If the half-life is 5 minutes, you'd have to pop a pill all day long. That wouldn't work. So we need a half-life that's a few hours so that it can be given once a day, twice a day, maybe 3 times a day. The fourth question cannot always be answered, especially in healthy subjects. It's, does the drug do what it's supposed to do? In our case, does it stimulate PKAN? Does it stimulate PANK? Sorry. And does it increase CoA? So we try to answer that question. And then the last question, number 5, is also sometimes not easy to determine, but we try. Does the drug go where it's supposed to go? In this case, does it go into the blood-brain barrier? Susie showed you that in mice it does. But does it go through the blood-brain barrier in humans? So to answer these questions, we do a first-in-human study. They're usually called SAD and MAD. SAD is a single ascending dose study, and MAD is a multiple ascending dose study. And the way we do this is we enroll groups of usually 8 volunteers. 6 would get active drug and 2 get placebo. In each of those boxes are 8 volunteers. So there's about 80 healthy volunteers that were studied here. They are in-house, in a phase 1 unit, under supervision, 24 hours a day, very closely supervised because it's a brand new drug, right? We don't know what's going to happen. Imagine doing this study in PKAN patients. It would take 20 years. It would be impossible to do. So whenever possible, we try to do these first-in-human studies in healthy subjects. I volunteered when I was a medical student to do studies like that. I got paid, right? So it's a win-win situation, and it advanced science. So whenever possible, we try to do these studies in healthy subjects. And that's what we did. The first group of healthy subjects got 3 milligrams, very low dose, single dose. Once we knew that this was well tolerated and everything looked okay, we gave the next group 10 milligrams, and we went up all the way to 120. When this was all good and it seemed to go well, we started giving multiple doses over 2 weeks. First group got 30 milligrams every day for 2 weeks, and we went all the way to 50 milligrams twice a day. That's 100 milligrams a day for 2 weeks. So let's find out if we were able to answer the 5 questions with this study. The first question, well, for 1 and 2, tolerability and early safety. Nobody's hair turned green. No bad things happened in terms of, nausea, vomiting, horrible headaches. We had a few side effects, and the most important one of which was, I'm so sorry, I have to get used to these buttons. The most important one is that in 3 out of 22 volunteers who got multiple doses, none of them in the single dose, but in the multiple doses, 3 of the 22 that got the active dose lowered their white cell count. It's called neutropenia. Our white cell count should be 1,400 per microliter or more. Well, 3 of them went below 1,400. The good news is none of them got in trouble. None of them got fever, none of them got an infection. As you know, white cells protect us from infections. So nothing bad happened because it was very short-lived. And when we stopped the drug, they recovered quickly within 10 days or less. So the conclusion here is that it is well tolerated overall, but it requires appropriate medical supervision. Does it get into the blood? Yes, it does. This, so sorry. At the end of the talk, I'll figure this out.
Well, and Mr. Gretler, or Dr. Gretler, I don't want to cut you off, but we are at time, so I want to be sure we have a few minutes for,
Okay.
for questions as well. So there's a 10:45 presentation that I know some of our folks need to be at. So.
Okay, I'll try to speed up a little bit. So does it get into the blood? Yes, it does. This is blood concentration over time. You can see that the more drug you give, you get the drug into the blood. It can be taken with food. We checked that. The half-life is 7 hours. That's very reasonable for once a day or twice a day administration. This is an important piece. Does it do what it's supposed to do? Does it stimulate PANC? Does it increase CoA? Unfortunately, it's very difficult to measure in the blood. We would have liked to do brain biopsies, but they wouldn't let us do that in healthy subjects. So we measured it in the blood. Very hard to measure in the blood, low levels, lots of variability. But let me take you through this unretouched, just raw data. That's the way science is. It's not always squeaky clean. If you look at the blue guys, this is placebo. 0 means that after taking the placebo, there was no change in CoA in the blood. If you look carefully, there's a number of blue dots below 0 where the CoA went down, and the same number of blue dots where it went up. So it sounds like it was a toss of a coin, pure chance. Some went up, some went down. But it shows you the huge variability. It's not something that's very constant if you measure it at various times. But it was symmetrically distributed around 0. Toss of a coin, pure chance. If you imagine an imaginary line at 100 nanograms per milliliter here, what do you see? You see that 3 subjects decreased, but everybody else increased. That does not sound like the toss of a coin anymore, right? That sounds like this is a real finding from the drug. But then what happens at higher doses, all of a sudden, people decreased again. Well, guess what? The scientists predicted this. Based on the exact mechanism of action of the drug, they told us that when we go to high doses, it won't stimulate PANC, it will inhibit PANC. And so that's what we see here. The good news is that there's a wide range here, maybe from 5 to 100 nanograms per milliliter, where there is an increase. But you see here an important lesson. More is not always better. Lastly, does it go into the brain? Those of you who had children might have had an epidural anesthesia where the anesthesiologist injects a lidocaine near the spinal cord to anesthetize the nerves. If you go a little deeper, you get the needle into the spinal canal, and you can get some CSF, cerebrospinal fluid. It's the fluid that surrounds the brain, and we can get a little bit of that. We found the drug there. The reason we found the drug there, it had to cross the blood-brain barrier. So the good news is the drug gets into the brain where it's supposed to go. So I'm going to go a little faster.
Is that maybe, again, I don't want to cut anyone off, we do have to get to this other presentation. Do we want to?
Can I show you one last slide? Yeah. So, yeah, sorry about that. I should have spoken more quickly. So we did a survey, equated a survey among 166 patients, caregivers, parents, and they were very helpful in telling us what works and what doesn't. Imaging's difficult. Okay, we got rid of imaging. Travel is difficult. We reduced the travel. This is essentially currently the design that we're discussing with regulatory agencies. A screening period of 4 weeks, that's normal. A baseline determination, a run-in to make sure the baseline is stable, and then dose escalation. We would start at a low dose to make sure it's tolerated, and then increase to the higher dose that we think is not too high, but that we think is the right dose where the CoA would go up. And that would be the double-blind period, 72 weeks. 57 would receive active drug, 28 would receive placebo. And you see there's not a whole lot of visits, 1, 2, 3, 4, 5 visits to reduce travel. That is the current design. So I'd just like to thank the conference organizer for inviting me. I'd like to thank all the individuals who participated in the survey, which is really helpful in designing a study that can be done, not just a perfect study scientifically that nobody can carry out. And then thank you to the NBIA Disorders Association and the NBIA Alliance for your continued collaboration in this work.
Thank you so much, and I'm sorry to have cut you off. Questions?
When is CoA going to be available in the United States?
I can take it then. Yes, thank you for the question. I don't think I can answer when it's going to be available because right now we are still in clinical development and it's a long process. So we are in the stages of planning for a pivotal study that's planned for early 2024. So once we go through the pivotal study design and discuss this with the regulatory agencies and they approve the drug, then it will be available. But it's a long process. I cannot answer specifically what date.
Yeah, so the drug that you're developing may be a good one and it may be effective in PKAN. We don't know that, but you said something that is wrong and I think it's important. I'm going to point it out. Could I ask the AV guy in the back to move the slides back to one of the earlier slides? Keep going, 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 keep going. Stop, go forward a few, another, stop. There is a single data point from Susie's lab that supports this in the mouse with a PANC2 mutation. We've done the study, and we don't see that CoA is decreased in the brain, and you have tens of data points that show that CoA is not decreased. And so this is not an accurate statement. And I think it's really important that you tell us, you convey true information when you're talking to the families. There's no evidence of a CoA defect in PKAN. It may well be that there is a CoA defect in PKAN, but there's no convincing evidence of a CoA defect in PKAN. That's really important for people to know. It may be a good drug. Chukri's drugs may be good drugs. They may impact PKAN, and maybe a trial should be done, but that's not an accurate statement. I think it's important that people know that.
Actually, there is a CoA deficiency in the early postnatal period in the PANK2 knockout mice, and then that goes away as the mice adapt.
We replicated that study of yours, and we did not see a CoA defect. And so it's we have
at 2 weeks?
Yeah, we have a different, and we looked earlier.
It's a lack of an increase because there's supposed to be an increase in CoA during the postnatal period, and that doesn't happen in the PANK2 knockout mice.
So we looked at control animals and the knockout mice. We looked earlier in the time frame you looked and later, and we saw no difference between the knockouts and the controls. So, I mean, like many scientific studies.
So how did you measure the CoA?
With the HPLC method, with a highly sensitive method, published method.
Okay.
I just want to comment on this point because it's a valid point. Our studies in yeast, again, I tell you, yeast is really fantastic organism. Many of you might ignore that. But what we are finding true, yeast has only one PANK enzyme. That's which makes the organism really attractive to study the basic biology. But we have mutants that have only about 10% PAN-K activity left, so we can really monitor everything downstream. We see absolutely no major change in CoA levels, yet downstream of CoA, we see it. So clearly something, the organism is compensating probably for the decrease in CoA level by bringing it from storage or something like that. So when you're measuring steady state levels of CoA, you might not see a difference, but maybe if you were to do this with labeled pantotenic acid to measure real-time production of CoA, you might see it. This is really a remarkable finding in yeast where we see a huge drop in the entire pathway, but we don't see the equivalent drop in CoA. Yet if we look downstream, for example, at the acetyl-CoA synthase and downstream steps, the change is dramatic. So there might not be contradiction here. There might be actually decrease in the pathway, but there are compensatory mechanisms either from storage or acetyl-CoA hydrolysis, releasing CoA, and what you're measuring on steady state does not reflect really what's happening at the pathway level. If that makes sense.
Could there also be an issue at the total CoA versus just measuring acetyl-CoA?
I think Susie's, and these are the experts here, it's very hard to measure the metabolites in this pathway. Yeah, it's from a metabolic standpoint, the cascade. Yeah, but acetyl-CoA can come from multiple sources, not just the CoA to acetyl-CoA. Yeah, no, I understand, but okay.
Thank you to all our researchers. You know, some of this is probably over many of our heads with some of the science, but we're so grateful for all the work that all of you are doing. And I invite you all back to Ballroom BC for the presentation of an award to one of our research facilities. Thank you so much.